So we are at the final speaker for today, which will bring us to the halfway point of the symposium. It's been an incredibly energetic and wonderful day. I hope you've all enjoyed it as much as I have. But I also know that you're going to enjoy our keynote speaker for today. And Kate Rubins, who I now regard as a friend, and I hope after spending a day with us, she will consider to also be a friend of NHGRI's. And I'm not going to read her bio, because I told Kate, you tell your story. I'm not going to read your standard bio. But I'm just going to say a few things in introducing her. First of all, for those who don't know, there are something like 42 NASA astronauts here in the United States. And Kate is one of them. And she also happens to be a biologist, an incredibly talented biomedical researcher, and is so interested in so many of the kinds of things we do at this institute. Now, you may be wondering, Eric, you're an impressive guy. How on earth did you get a NASA astronaut to come here for an NHGRI symposium? And that's a reasonable question. And it turns out that it happened as follows. When Kate was up in the International Space Station, which I'll tell you more about, in 2016, one of the things you get to do, I have come to learn from Kate, is that you get two celebrity calls, where you get to be on the phone and have a conversation with celebrities. You get two of them. Now, the reason that Kate and I have gotten to be really good friends is, like me, she's an incredible nerd, okay? So much so that she used one of her celebrity calls, not on Taylor Swift, which is what I would have used, but rather she used it to call Francis Collins. Okay, so I didn't say she had great judgment. I just said she had nerdy judgment. And so she and Francis talk. She's up in the air, he's down here, probably in building 1. And during that call, Francis invited her to come to NIH the next year for DNA Day. And that was 2017, and Kate came here for DNA Day. That's actually when I first met her. In fact, we all gathered in building 1. We used to do active chat rooms on DNA Day, where high school students and others would send in questions, and we would answer them live, and Kate was there answering, and I was there for answers. So we all did this big DNA Day chat room together, and I also met with her, and it was wonderful, and I got to know her. Fast forward, of course, then we had the pandemic and all these other things, but meanwhile, Kate was back up in the International Space Station, actually during the pandemic. Once again, she gets 2 celebrity calls, and once again, she wastes one by calling Francis, okay? I don't get it, but okay. So again, keeping in communications with us. But then, pandemic ends, and we are getting back to normal with these professional meetings, and I am one of the co-organizers of the LGBT meetings in Florida, and I said, we've got to get Kate to come and give a talk there. And so earlier this year, in February, she was one of our keynote speakers, and as she's done today, she was a full participant, and really was wonderful to have her, and we spent a lot of time talking then. But her talk was incredible there, and I just thought to myself immediately, we have got to share Kate with the rest of NHGRI, because it's just so enjoyable to have her, interact with her, and hear her speak. And so that's when I put in the plug, and I said, we have this annual symposium, would you please come be our keynote speaker? And she said yes, and the rest is history. So that is all I'm going to say. That answers the question, how did I get Kate Rubins here? And she's going to tell you more about herself and tell you about her experiences as an astronaut and as a scientist. Kate.
Thank you. Thank you so much. That was great. I appreciate it. Hi, everybody. I am really excited to be here. I've actually been looking forward to this, I think, just as long as Eric has. So you guys know already that I'm a complete nerd. It's fine. I own it. It's all right. And so I got invited to come give a talk here, and I've just been like giddy about this for the last few months. So I decided I need to have a really cool talk title, right? So the best I could come up with was Genomics for an Interplanetary Future. I'm not sure we're going to quite get there, but at least it's a start. So he said, come tell us a little bit about yourself. And so I'm actually going to start in the middle of the story. So this is me in 2016. This is the Soyuz capsule that we launch into the International Space Station. So I'm in the right seat. I get loaded in, the left seat, and then the commander. And so my commander actually took a photo of us just before we were getting ready to close the hatch. And I look happy here, but really we have about 4 hours to sit on the launch pad before we launch, and there's not a lot to do. And that's a lot of time to think about how I got there. What were all of the very bad decisions in my life that led up to this point, sitting on top of what is essentially an ICBM getting ready to be blasted into outer space? So, as I'm pondering of all this, I'm gonna then start at the beginning of the story. So this is my bedroom wall growing up, and everybody who is an extremely specific vintage in time is gonna get distracted by the 90210 poster on the right. And the rest of you can take a look at what I thought was super cool. Like I said, I'm a nerd and I own this. But when I was a kid growing up, I wanted to be an astronaut, a biologist, and a geologist. And I was going to do all three of those things, of course, because you can just have 3 careers. I went to space camp, back with the nerd theme. But then I actually started getting really interested in public health. And my dad was a social worker. He worked for the Napa County Public Health Department. And he got me involved in high school in a program that did peer HIV education and outreach. And so as part of this program, we had physicians, we had clinicians come in, we had patients come in, and we had researchers come in. The idea being that the students needed to be the most educated about the subject matter that we were telling our fellow peers. We needed to have accurate scientific information, and we needed to have the patient perspective, and we needed to have that clinician perspective as well. And so we spent a year first learning about HIV, then putting together a production, a show, and then bringing this around to the other schools for HIV prevention. And so as part of that, it was an incredibly impactful experience for me to hear from patients, and this was the mid-90s, and HIV, while we had protease inhibitors at that point, resistant strains were evolving, and it still could be a debilitating disease pretty quickly, and it could be a death sentence. And so talking to the researchers and hearing about what they wanted to work on and how they were trying to prevent this disease from progressing, and learning about antiviral resistance and how it integrates into the genome as a high schooler, this was just incredible. And I said, this is what I want to do for the rest of my life. So I went to undergrad. I worked in the HIV Research Institute, the Infectious Disease Lab at the Salk Institute, and studied integrase inhibitors and how HIV integrates into the genome. Was fortunate to have the wonderful mentorship of Rick Bushman there, and then went to grad school and decided that I wanted to study some additional viruses. And so I started studying Mpox, which it was a pox virus. Nobody knew about it at that point. We're all a little bit more familiar with it, unfortunately, now. And then September 11th happened. And they said, you're off of Mpox. We're studying smallpox now. And so I started working with researchers at USAMRIID, which is in Frederick, Maryland, just up the road in the Biosafety Level 4. We started collaborating with the Center of Disease Control as well, and going down to Atlanta, and it was kind of like all guns a-blazing. We really needed to work on smallpox vaccines and therapeutics. And when you meet the smallpox people, then you end up getting to know the Ebola people and the Marburg people and the Lassa people. And so pox viruses and filoviruses were kind of what we did. And so this was really it was my Ph.D. thesis, working at the Center for Disease Control, and I got a chance to work with NIAID as well, and USAMRIID, and the Defense Threat Reduction Agency. And so this was such an exciting time to be able to work on all of these things, that when I was getting ready to graduate, I said, I just want to keep doing this, but I also want to be determining what I'm working on. And I wanted to work on Mpox back in the day, and I really didn't feel like enough work had been done on that. And so, at the age of 26, when I knew everything in the world, I thought, well, I'll just start my own lab, because that's going to be, that way I could work on whatever I need to work on. And there were 3 fellows programs in the country where, as a recent grad student, you could have PI status, and you could get funding, and you could start your own research laboratory. One was at UCSF, and then 2 were in Boston. So I did not have a job, but I moved to Boston because it was statistically most probable that I would get into a fellowship program there. And I also figured, in Boston, there's postdocs all over the place. Like you can find a postdoc, so it would be okay if Plan A didn't work very much. And so I started at the Whitehead Institute, and built a research group there. And we started working on Mpox. We were looking at genomics of poxviral and filoviral infections, both from the virus side as well as the host immune side of things. And we were very interested in animal models as well as therapeutic and vaccine development. And as part of this, I actually got a chance to do some research, some field work with a team that was going to the Democratic Republic of Congo, because this was the only place at that point where Mpox was found in nature. And it had kind of bothered me a little bit that we did things in the lab with these strains of virus, and we'd passage them, and we'd say, oh yeah, you know, this Mpox or this Ebola strain. And I'm like, well, how long has it been living in the lab? And is it really the original thing that came out of the patient? And so one of the things that we wanted to do was actually look at primary clinical samples and get really good data about that. And then as part of that, and I'm a Ph.D., I'm not a clinician, but as part of that, we had an incredible partnership with clinicians that were working at the National Research Institute in Kinshasa in DRC, and we had physicians from DRC that were working with us to go out to the field, and so they would provide a clinic for the patients, and then we would do the informed consent. And I really liked Liz's briefing earlier, because that informed consent in the Congo is a huge challenge in making sure that we are not coercive in any way possible. And I actually considered one of the big successes of that study when we had some patients that refused to consent and still got their care for themselves and their families. And we realized, you know, hey, this system is really working. You know, we had some great clinical outcomes, and it was really, I just felt like I was doing what I wanted to do my whole life. That being said, it's very challenging to work in the Congo. You have to transport your supplies in, you know, really interesting makeshift ways. Do whatever you can to get the medical equipment out there, to get the lab equipment out there. But this is not doing lab work in anywhere that looks like a hospital, right? So this is my biosafety level 3 that I set up in a supply closet at the hospital with no air conditioning. And trying to figure out a way to make all of this work was incredibly challenging, and I'll talk about this a little bit later. So back to the lab. I'm a researcher. I'm doing what researchers do, which is apply for grant funding. None of us like it. And I was working with a very long-time collaborator, Lisa Hensley, who actually used to be deputy director at the IRF, the Animal BSL-4 facility here. And we had worked together since I was a grad student and she was a postdoc. So we'd collaborated on a lot of grants, and we were writing an NIH grant. And we started late. We weren't very on time about this. You know, scientists, we never are. We were a bit behind. We'd been staying up really late nights. We'd been working on it, and the grant kind of sucked. It just wasn't a very good grant at that point. We couldn't figure out what we were going to do. Specific Aim 2 was giving us all kinds of trouble. We hated ourselves, we hated our lives, we hated the grant, we definitely hated NIH at that point. And she called me up and she said, this is going terribly. I think we're going to have to get new jobs, and let's get a job where we don't ever have to write a grant again. So I was looking at usajobs.gov. And on the front page, there's applications to be astronauts online. We should apply to be astronauts, because then we don't have to write grants anymore. I said, sure. We hung up, and I didn't want to work on Specific Aim 2 anymore, so I applied to be an astronaut, as one does. I called her the next day, and I said, Lisa, I got my application in. Like, you know, did you get yours in? And she said, Kate, sometimes you have the problem where you don't understand where somebody's joking. That was a joke. And I said, well, don't worry. You know, NASA's never going to call a biologist. They've got rocket scientists and engineers and everything like that. And as it turns out, be very careful what you apply for. Because about a year later, I started in the 2009 astronaut class, which is the series of bad decisions that led me to being on top of the rocket. But what I really wanted to talk about today is what we do at NASA, the interesting research that we do and how that relates to research on Earth, how I think the space station can actually be a very interesting forcing function to study human health and disease on Earth, and how it can be a toolkit for how we think about designing research and designing devices. So this is the International Space Station. I thought the view of the Pacific Ocean at the Salk Institute was amazing for a laboratory view until I got to the space station, and you can see the planet as you're doing your research. It's an incredible machine. It's the biggest engineering achievement, I think, that humanity has come together to do. This is the International Space Station, and we have built this together. It's been orbiting the Earth for more than 20 years. This is really a symbol of our peaceful international cooperation in outer space. I just want to tell you a few stories about the kinds of science that we do in space exploration. So I want to talk a little bit about DNA sequencing in space. Like, you guys are my target audience for that. You're going to really get it and have fun. I wanted to talk about the water purification that we do, some sustainability, recycling, sensing, all of these kinds of things that have a genomic aspect to them. And then also human health monitoring and what happens to astronauts in space and how some of those dysfunctions parallel diseases that we have on Earth and ways that we can study diseases on Earth. So first up, DNA sequencing. So why do it in space? Really, the driver of this was microbial and environmental sensing. We have astronauts on the space station. We have a very interesting biome up there that has been separated from the Earth for more than 20 years, that has regular injections of dirty humans that come in, but only 3 of them, and then they leave, and then we get some more humans up there. So it's kind of a really interesting, it's a closed environment that's very well controlled, where we've got very specific and very monitored humans that come up there. So of course, this is like a fascinating research project. But really, the main driver was if we have a problem, if we have a microbial problem on board the ISS, we have to wait often up to 3 or 4 months until a vehicle is ready to undock from the space station and land back on Earth, because we don't have a clinical microbiology laboratory up there. So we can take a sample, we can swab a plate, wrap them in parafilm, put them in a space vehicle, hurtle them back at the Earth, land on Earth. We would send a plane over from NASA to Kazakhstan to pick up the samples, fly it back to Johnson Space Center, and that's finally where it gets to the lab. So you're talking up to 4 months to diagnose something that's going on in the environment or in a human. And as we all know, bugs can grow a lot faster than 4 months. And so this was a risk for us. This was a problem. We thought we would really like to have a technology where we could do point of care diagnosis or try to understand what was going on with microbes on the space station, particularly given that it's a closed environment where our air and our water is recycled. Some kind of microbial outbreak could be a big problem for us. And that kind of platform would then allow us to do other things. We can profile all the very interesting physiologic changes that happen as a human adapts to microgravity. We could monitor human health and disease much closer to real time. And we could increase this Earth independence as we're going, to these lunar missions and these Mars missions where we're not going to be able to send samples back to Earth. We're going to be gone for 2.5 years on the way to Mars, and we have whatever lab we bring with us. And then as we get really into the science fiction, astrobiology and detection of life. This is one of the reasons that we're going to Mars. We're interested if there's life on Mars. And so sequencing, ways to look at non-canonical bases, and ways to do life detection is of great importance to NASA and to the space agency. So we really started with this microbial sampling idea. That was the main driver. And so we wanted to, eventually our goal was to do swab to sequence. But we had a lot of challenges with this. We had never done sequencing on board the ISS, and it's the most incredible engineering marvel, but it's actually very rudimentary sometimes when it comes to our capabilities, because it's very hard to get equipment into space. We're space limited. We are very limited with our cold chain, with consumables. We can only send things up and back about every 3 or 4 months. And so we had this issue of power, mass, and volume. The benchtop sequencers that are this big are just way too big to send up the space station when it's competing with all of this other equipment. We don't have the space for it. We don't have the power draw available. We also had a pretty big problem with liquids in microgravity, and just in general in our laboratory setup. And so you guys all know what the sequencers look like and sort of their size footprint. The things that have lasers and CCD cameras and fluorophores are very tricky and challenging to maintain. There is no technician that you can call in space. The astronauts have to learn how to do it all. And by the way, it also has to survive a rocket launch, which I can tell you has a lot of vibration going on. There's a lot of G's there. And so the standard equipment that we had just really hadn't been practical to send onto the space station. Then nanopore sequencing came along, and something that you can fit in your pocket, that, if the sequencer is not working, you just pull another one off the shelf, and had a consumable footprint that was pretty low. Seemed like a technology that was going to be pretty well adapted to space. However, we didn't know if it was going to work. And one of our main concerns was actually the liquid handling in microgravity. So if you don't have gravity, essentially the forces of adhesion and surface tension dominate. And so this is really freaky when you get up there and you can hold a ball of water in your hand, and then your crewmate's like, oh, put it in your eye, because they're all fighter pilots and that's what they do for fun. And you put it in your eye and it kind of creeps across your face, and it's just like a horror movie. So it's really interesting what fluids do in microgravity, and they will stick to whatever surface you provide them with. And they'll stay there, and they'll just kind of spread out to the physics limit unless you give them something that's a little bit more hydrophilic and move them along, and then they'll move to the hydrophilic surface. So if you provide them some motive force, they will move, and the way that you get that off your hand is to pretty violently jerk it, and then the water sprays all over the ceiling, and then you got to go clean it up so it doesn't get into any of the equipment. Close personal friend of mine. So these are our challenges with lab equipment, which has really been designed with gravity in mind, because why else would you design a piece of lab equipment on Earth? And so one of the things that we started out with was just let's figure out what's going on with fluids in space. And when I started at NASA, I came in and I asked about all the laboratory capabilities, and I was told there's very little laboratory capability on board the space station for molecular biology because we don't really pipette in space. And I asked about that. I was like, well, tell me more. And it turns out we designed experiments to be shuttle mid-deck lockers. So these were like a shoebox size piece of equipment that would be designed on the ground. It would be this very specialty equipment. It would go up, you would fly it on the shuttle. The astronaut would start the experiment, push a button, and then we always joked, is there a treat that comes out the other end, for the monkey if they push the button? Because that's essentially what we were doing. And we didn't do anything. We didn't do open container. We didn't transfer liquid anywhere. And really the response I was given was, well, astronauts probably can't pipette. It's too hard. And by the way, that's just not going to work in microgravity. You can't do open container stuff. And at that point, I didn't know about this adhesion and the surface tension. But it didn't really make sense to me. I was like, I can't get a good technical explanation out of you people as to why pipetting won't work in space. And that made me really mad. So on my first flight, we have behavioral health support, and we have a team it includes a psychologist, and we get to pick some things that are going to give us psych support, and our celebrity calls are part of this psych support kit, and as part of it, we get about 5 kilograms of up mass to the space station. And so people usually, they bring brownies or cookies and letters from home and stuffed animals and all these sorts of things. And I'm kind of a practical person. So I brought a couple photos and some letters from home. And then I filled the rest of my psych support equipment with scientific equipment. And I got the psychologist to sign off on it after about a 20-minute meeting where I said, look, I'm a scientist. You know what makes me happy. You need to sign that this is going to be good for my mental health when we're up on board the space station. And he was definitely on board with this. And so I just these are things that I brought in my support equipment along with some food coloring. And I tried everything from a 384 well plate all the way up to a 50 mil conical. I brought different kinds of plastics. We really defined when we're looking at an Eppendorf tube or a 96 well plate. What we discovered was that there's just this meniscus goes a little bit higher in the plate, and so you have to account for that, and you have to use filter tip pipettes. So we also tried positive displacement pipettes, and you just pipette a little bit less than you normally would on Earth, and the whole thing works. That the volumes that we're talking about, the amount of plastic that we're talking about, really holds these liquids in space. And I can be snarky when I want to. So I just took this video, I sent it back down, I said, pipetting works. And it's great. It's launched this entire cottage industry of open plate experiments that we can do. There's other things that are difficult, right? We didn't have a centrifuge. So we 3D printed a rotor, we stuck it on a drill, and now we have a centrifuge. This is what it's like to do experiments kind of in this challenging research environment from space. And so we put all of this together, we put it on a nanopore sequencer, and when we sent this up, we said, we don't expect this to work, actually. We don't think this is going to work the first time, but it's going to be interesting to see where it doesn't work. And where it breaks is going to help us focus our engineering and our scientific efforts. And so we sent up prepared libraries. They were prepared on the ground. They were all ready to go. We split them in two. We sent half to space, and we kept half on the ground. And we put in the sequencer, and this was like my most nervous scientific moment in my entire life. Forget holding 10 to the 9th of smallpox in a vial. Having this library in space and having hundreds of people watching me, I didn't want to squirt the DNA library and have it be flying around the space station. That needed to go right into the sequencing cell. So luckily, thank God I got it right, and we did the first sequencing. And to our surprise, it worked. It started, spitting out base pairs right away. We sent the data back. We compared it to the sequencing libraries that we had prepared on the ground, and it worked. And it worked the first time. And I'm not the bench. Never in my life has something worked perfectly in the laboratory the first time. But if you're going to have that moment, it's really good to do it on the International Space Station. So we did a whole study on microbial sampling on ISS, and what we did was we did a mixture of Lambda, E. coli, and mouse genomes. We wanted a range of GC content. We wanted to prepare the libraries to make it easy enough that we weren't troubleshooting that part of the experiment, but we wanted those libraries to be complex enough that if we did get it to work on space station, we could prove some things like we could pull out different species from this mix and get that nice GC content. So we were able to do that. We were able to reassemble genomes, and that all went very well. And so from there, we've gone on. So we then decided we wanted to do the sample prep, the library prep on board. And so we tried taking DNA that had already been lysed and prepared, but then we did the library prep on board the ISS. And then we went to do the swab to sequence. So we went all the way from a sample that we took. We did streak on a plate, picked a colony, sent half of it home, and did the rest prepped all the way through. And then the part that we sent home got sequenced down on the ground, and then it also got identified by classical microbiology methods, and that was cultured off Earth, and it worked. We identified the right things. So we've done direct RNA sequencing, cDNA sequencing. We did this culture-independent swab-to-sequence profiling, and then there was a really cool student that was actually, this was a student project, who decided to do a CRISPR-Cas9 genome detection and editing and detection. And so we've demonstrated all of these things, but as you guys know, this is a platform technology, and so there's a lot left to do still. And I'll talk at the end about some of the things that I think we could use this technology in the future. So next I want to switch to this water purification, sustainability, recycling, and sensing. And so NASA, I think, has one of these incredible success stories, which is our closed-loop water system on board. So this system is 97% closed-loop efficiency. So that means the water that's up on the space station has been there for decades. The water that was on my first flight was on my second flight still. And the way that we provide this water is we have a system that takes all the condensate out of the air, and then it takes all the liquid waste from the humans, puts us in this dirty water tank holding facility, and goes through a process of purification that then turns it back into our water for drinking and for rehydrating our food. And you can see here the equipment is really big. It's like 3 refrigerator sizes to do all of this water purification, but it works. And of course, it freaks the astronauts out. You tell them, hey, you're going to space, and you're going to drink your pee and your crewmates' pee, and it's going to be all great when you get up there. Nobody likes thinking about it, but I've tasted it. It tastes great. It's not like the Saturday Night Live skit where they're spitting it out. The water is fine. But the fact that we just have these same water molecules for a decade and we're keeping these in these systems is incredible. This technology is really incredible. So I think this is a huge success story from NASA. And there's a lot of things that we're thinking about in the future. Those are very big systems, so we want to get these down to smaller, more portable, and more modular systems. So we've tested aquaporin filtration on the ISS. We've looked at nanoparticle-based fibers for disinfectants, potable water disinfectants, non-chemical biocides, and then coatings to prevent biofilm formation in our recycling systems and pipes. And all of these are active areas of research. One thing that we've kind of tried recently is sensors and taking a look at what's inside this water system. Because of course I'm a microbiologist and I immediately want to know what's growing in this system, because probably something is. And the way that we've been sensing it so far is we just have a machine that detects total organic and inorganic carbon. We're basically measuring the organic carbon as a proxy for a bioburden in this water system. We also use iodine as a biocide, and then we filter that iodine back out before the humans drink it. But this wastewater tank has minimal microbial control, except for the iodine and monitoring, and we regularly monitor the water for a couple different kinds of, like massive levels of bacteria. We'll just put it on a filter, put it through a filter, and we'll look at colonies. And then the NASA scientists say, okay, well, if there's 10 colonies or less on there, it's clean. And if there's 100 or 1,000 colonies, it's not good. Call us. We need to do something. And that kind of drove me nuts, too, because I'm like, well, it could be fine. There could be 1,000 colonies of something that's perfectly fine, that's safe for humans to drink. And if there's 10 colonies of something that's really bad, then the water's probably not okay. So we're working now on metagenomic methods to look at different parts of this spacecraft water system to see who's there, who's growing there, how does that change over time. And we don't have the answers yet. These experiments just started taking samples this summer, but I think it's going to be really cool, and it's definitely going to freak out the NASA engineers because they're going to discover that things are in their system, and there's going to be some panic and some safety meetings, and then we'll all get back to a good place. But this has really gotten me thinking. We've gotten this completely closed-loop water system, and water. We use the water for hydrolysis. So we split the water. We use the oxygen to breathe. So we regenerate our own oxygen there as well. We haven't done this for other molecules. And so if you're thinking about, let's maximize sustainability, let's reduce our waste, we really are incentivized to do that on the space station and to figure out how to do that, to spend the resources to push that engineering forward, because it is so expensive to launch things from the ground. And so that pushes us in an engineering direction that we wouldn't otherwise invest money in kind of trying to solve these engineering problems. But waste is not free. And we don't really tax waste in this country, and we don't pay attention to how much waste we generate. Obviously, we're now starting to wake up to this, and we need to do this more. But we have a system where NASA's counting every gram that you launch into space. So what a great place to start thinking about sustainability and actually looking at every single molecule of waste we generate and what we could do with that. So right now on board the spacecraft, we capture the CO2 out of the atmosphere. We still live in much higher CO2 levels on board the space station than we do terrestrially. So it can be 1,000 times higher than terrestrial CO2. So we capture the CO2, we put a little bit into a Sabatier reaction, but then most of it we dump overboard. So we're just trashing this CO2. But that's got carbon. Carbon is useful. So could we couple that with the synthetic biology system that's then taking that carbon for microbes? I also told you about how we kind of filter the water. We got a bump in our efficiency of our closed-loop water system when we started taking that this kind of a brine sludge, and we spread it on we developed a piece of equipment. We spread that brine sludge out, and then we evaporated that. By blowing air past it and evaporated all the water that we could recover out of that last bit of brine sludge. And funny story, the place that they installed it is like right over the exercise equipment. And I was in the crew that put this in, and it did not smell good. So your whole time during your workout, it was like doing a workout in a urinal somewhere. And so we had some comments to the ground. We gave them our feedback, and we worked really quickly to get some filters on this brine diffusion experiment. And so, what are the other things that we then throw away all that dried brine? We just pack it in a spacecraft, we burn it up in the Earth's atmosphere. But that's got a lot of nitrogen in it. It's got phosphorus, it's got other cool things. So you could feed those nitrogen to plants. Then that can produce food for us. It does a little bit of CO2 scrubbing. We're not going to have like the biggest massive plant facility, but it helps us out there. It generates oxygen. It can potentially be useful, again, if you're having a synthetic biology track here for the microbes, and probably a lot of other useful, interesting chemicals, and then probably a lot of chemicals you don't want that you're going to get rid of. And then so lastly, we do have this Sabatier reaction. So we take a little bit of the carbon dioxide plus some of the hydrogen that comes off of our water splitter when we're doing hydrolysis. And then we use this for additional water recovery to pull out a little bit more water. But the methane is discarded, and that's also super interesting as well. Methane can be fuel. This is really important in spaceflight, fertilizer for plants, all kinds of other things. So really the big question here is how much is the juice worth the squeeze? How much effort do we want to put into recovering all of these different molecules? But NASA at least wants to try. And we want to try to figure out ways that we can get the most out of all of these different molecules, closing our loops in all of these different systems and figuring out how we can have the HVAC system that you live in and the engineering and these mechanical systems start to interface with things like plants and with microbes and with synthetic biology to provide a clean environment for humans. So I'm really excited about this going forward. And then I also want to talk about human health monitoring in space. So we don't have much of a lab in space. We don't have much of a hospital. We don't have any ability to do MRI. We can't do X-ray. We can't send somebody to a CT scanner. What we've got is ultrasound. And let me tell you, we use ultrasound for anything and everything that you could possibly use. We get very good very quickly at ultrasounding all these different parts of our body because that is the imaging capability that we have. And we want to use this both to prevent dysfunction in space, but also as a way to understand what happens to the human body in space. And so a lot of the medical experiments on space station, the human research, is seeing how our human systems adapt to a very different environment, which is microgravity, and then also radiation. And so astronauts experience this whole spectrum of adaptation in flight and post-flight. And it's not actually that we are dysfunctional in space. It's that our bodies adapt so well, so quickly to microgravity, that when we come back to the planet, we are just maladapted to living in gravity. So when you get up there, there's this period of about a week or two where things are going haywire. I mean, you really feel terrible. All your systems are just really messed up physiologically, and you don't know what's going on. And then after about 2 weeks, the body, everything comes together, and all of a sudden you're well adapted to microgravity. And so I'll just tell you about a few of these different systems. So neurovestibular. When I launched to space, and I first finished all of the rocket staging and get the solar arrays out and check that we have a good atmosphere, and oh my gosh, check our body parts, we're not actually dead, we appear to be in one piece. Then you got a chance to okay, how does that feel being in microgravity? And it feels like I have just been kicked down the stairs continuously for stairs that never end. That's that sensation that you have of just falling and tumbling, and your brain's going, I don't know what you've done to me, but it's not right. And that neurovestibular, that kind of maladaptation persists. It can go as long as a week. Most people usually adapt within about 3 days of understanding. The brain kind of starts to understand what's going on. We have the same issue coming back. There's a lot of neurovestibular dysfunction coming back, and people are studying this. It's a really interesting way to study this because you've got a normally otherwise healthy adult where you can turn it on and then turn it off again. So you can provoke the system and then study what's going on. My kind of hypothesis is that it's how your brain is responding to signal input. So when you first get up to space, we learn physics all over again really quickly. So you float over to the laptop and you want to send an email and you start typing and you hit the keyboard and you will very slowly make your way to the ceiling. The first letter that you type. We have to hook our feet under some handrails and learn how to stabilize and offset all of these different forces. When you are trying to turn, like you just turn a little bit and you will turn and 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 never stop turning unless you hit something else. And so your brain's not used to this. And so this tiny amount of force is provoking this huge change in direction, vector motion. And so I think this is mixing you up. This is confusing your signals, and so this is kind of like what happens when you're car sick or when you're motion sick. These are, by the way, this is all Kate's hypotheses. I'm not actually a neurovestibular researcher, but this is just what I've learned from being in spaceflight. Same thing when you come back to Earth. You expect that you will have you do your tiny little input, and then your brain's like, well, nothing happens, and I expect to be on the ceiling right now. And so there's that mismatch, and there's that that dizziness, the vestibular, just malfunction. I mean, it's terrible. You just really don't feel good. The cardiovascular system is super interesting. The heart becomes a little bit more circular. We were worried, and we still are, about arterial stiffness, which mimics some aging. And so we were doing lots of ultrasound on the arteries and trying to get all this imaging and collect all this data. And this is actually published as a New England Journal paper. We were doing this ultrasound on an astronaut and discovered a giant clot in the jugular vein. And this is no bueno. People were incredibly concerned. We spent a lot of clinical time and attention on this and followed the clot. And this is concerning, obviously there's a big thrombus there. We're concerned about that. But then if you put somebody on blood thinners and you have to do an emergency undock from the space station and you slam into the planet at whatever G's we're pulling, you could have a lot of trauma. You don't want somebody on blood thinners for that. They went back and they looked at previous, because this was purely by coincidence that we discovered. We were doing a different payload, we were doing a different research study, and we found this clot. So they looked at wherever they had data where they thought that they could see this, and they actually found, I think it was about 4 out of 10 of these subjects had some evidence of thrombi. So what that led to was a new discovery, and this is just in the last few years, that astronauts can have stagnant or even reverse jugular blood flow. And that's probably contributing to this clotting buildup. And so we're testing people for clotting dysregulation now. We're ultrasounding our venous system all the time, seems like every other week, and we're trying to learn a lot more about this. But this kind of shows you, we've been going to space for a while, but we're still discovering what happens to the physiology. Probably everybody knows that astronauts lose bone. This was a huge problem for the space program. So we would have in 1 month, an astronaut would lose what an 80-year-old osteoporotic woman would lose in a year of bone loss. It was such a problem that in the early days of the space station, our water filtration system was getting clogged up from all the calcium that we were excreting into the water system. So this is a success story. We've really gotten a hold of the bone thing. What we do is resistive exercise. And so we have a giant set of vacuum cylinders, and it's attached to a squat rack. And so we do things like squats and deadlifts and crunches and all of these sorts of things. And we have to do exercise for 2.5 hours a day, every day, mandatory, just to keep the bone that you launched into space with. But we have a countermeasure. We have figured out how to do this. And so we're still studying this actively because it's hard to find another analog of a healthy population that healthy 40-year-olds that have full bone, great skeleton, and then all of a sudden, boom, they're losing it very, very rapidly. And then if they've lost it when they come back to Earth, what we really find is when they build it back up again, if you've got that trabecular structure in your bone and you lose it, and it's not touching, it's not going to build back up again. So we can quickly rebuild bone mass in astronauts, but it all builds up in the cortex, and so that affects your tensile strength. So this is still an active area of research. We have something that kind of like stop the bleeding, but we need to figure out more about this, and we're learning about human physiology. We know that we lose a lot of muscle. We exercise, that helps a little bit, but we still lose quite a lot of muscle. So when we come back, we have to do very intensive physical therapy for 2 hours a day for at least the 30 days that we get back. We know that there's immune dysfunction, so this is super interesting. There actually have been reported cases of shingles on board the ISS in normally healthy, standard immunocompetent people. We started digging into this a little bit more. We started seeing EBV reactivation, of course the VZV reactivation, and all kinds of other things. We studied the immune system, and we saw dysregulation there. Interestingly, we also see this kind of dysregulation in people that overwinter in Antarctica. And so we're trying to tease apart now, what parts of this are due to stress? Like, very stressed out when you're in space all the time. We're working 12-hour days, 14-hour days sometimes. You're supposed to have a day and a half off on the weekends. They almost always take that away from you. You're working your whole weekend. And by the way, you're in an environment where if a grain of sand penetrates the hull, you can all die pretty rapidly. There's a lot of stress going on the space station. But there's also a lot of physiological stress and mental stress for people that are overwintering in Antarctica. They're separated from their families as well. They cannot go home if there's a medical issue. It's dark all the time. They're confined in this environment. And so it's going to be very interesting to see where the microgravity and radiation effects play in versus just this overall kind of system that we've constructed that put humans under incredible physical and mental stress. Nutrition is definitely an issue on board. Kind of the cool thing about nutrition, and I participated in one of these studies when we first got our food tracker up there, is that we actually know every single morsel of food that the astronauts put in their mouth because we record it in this food tracker. And it started out as a study where it's like, you sign the consent form and then you have to participate. You have to track your food. But it became such a cool and fun thing to track your food that all the astronauts do it anyway, whether or not they're involved in the study. And so we've got this great metadata on what people are eating. We know that nutrition is going to be a huge problem on the way to Mars because we're going to have to provide shelf-stable food that's stable for 5 or 10 years. All these great antioxidants and everything are pretty labile. They get broken down by the massive radiation that we have in space and temperature swings. And so we're really concerned about the level of nutrition that we're able to provide when people are in this challenging environment for a long time. Behavior, it's always interesting, lock 6 people in a tin can and see what happens. The real issue is not usually the astronauts on the space station. It's the interaction of the astronauts on the space station with the ground controllers. And I experienced this as well. So you're up there, you're working 12, 14 hours a day, you're stressed, you can't see your family, you are in this enclosed space all the time, and the ground is always telling you what to do. They plan your day in 5-minute increments. And there's a red line, the red line of death that just marches through the day that tells you where you're supposed to be, and you're always behind that red line, and you're just always trying to catch up to this red line. So that can be incredibly stressful. And of course, the people on the ground, they're people that I know, they're my coworkers, we're friends, they're good people, they have good intentions. It's amazing when you are separate from that group of people and you're under stress, how quickly they become the enemy. Those idiots on the ground do not understand what it's like to be in space. I can't believe they're telling me to do this. This procedure is messed up. How can they make me make so many mistakes in my day? And by the way, they get to go home at the end of their shift. We work 3 shifts in mission control, and I'm working 2 of them on board, and I don't get to eat any cookies. I don't like them. And so this is really interesting. There's a cool set of astronaut journals that's been published in a psychology. It's called the Astronaut Journal Experiments, and this has been published as a paper, and you can go read up on it. And this the astronauts kept diaries, and then it was analyzed for sentiment analysis. So this is really interesting. I learned a lot about human interaction and how much energy it takes to maintain positive interaction between 2 groups who are starting to have kind of diverging realities. What you need to do to make sure that those groups stay harmonious. And so one thing I did with my crew is I really asked them to stay positive. And I fell victim to this too. I'd be bitching about the ground all the time, and I'd catch myself and I'd say, what, we have to assume positive intention. We have to say nice things about them and nice things about ourselves. We're all gonna work together and get through this, but it actually makes a pretty big difference. We did fun things like cookie decorating contests towards the holidays. I made little costumes out of whatever bits of fabric we had left over, and we all wore silly costumes. And then a flight director, we had a costume contest, and a flight director dressed himself up entirely in wrapping paper and sent us a video, and he won, absolutely. And so I was kind of chuckling when you guys had your video presentation and your shenanigans kicking this off today. That stuff is important. It's really important. It's good to have fun together and to bond as a group. But this is a fascinating, psychologists love this, fascinating to study this. And then, of course, radiation. And so we are looking at all of these systems in response to spaceflight, and we're doing this both from the human research study side, where we're collecting clinical samples, as well as the in vitro side and in tissue culture. So one thing I just want to touch on really quick was how to do cell culture on board. And so again, before my first mission, we really hadn't done long duration cell culture. The longest we had done were a couple-week experiments on shuttle. And again, the explanation was, it's too hard, astronauts can't pipette, they're going to contaminate everything, and we just, we don't have the resources to do this. And so we worked really hard with the lab to figure out how to do cell culture on board the space station. What we had to come up with was a system that looked like a tissue culture plate, but then had a gas-permeable membrane and lure lock ports so that we could put the media in so that we could change the media on these cells. And this was an incredibly laborious process. Like normally you go in, get your 6-well plate, you're done. It would be like an hour that I'd still be exchanging things with a syringe and making sure I got it all well on the plate. And so we did this for 30 days. And let me tell you, every time I changed the media, I was petrified that I was going to contaminate these cells and ruin this experiment and think that, like again, if something's going to work, let it work on the space station. I didn't contaminate everything, so whew. So we were looking at cardiomyocytes on the ISS. So we had induced pluripotent stem cells, which we differentiated, split half on the ground, split half onto the space station. And then we were doing video microscopy and image analysis. We sent some back so we could do live, so that we could do contractility analysis, calcium imaging. We fixed some so that we could do some additional imaging of time points during the time course, and then we did gene expression at various time points during this time course. And this is all published. Here's a cool picture of like inside the glove box that we had to build. We had to build a BSL-4 glove box because safety was so concerned about the cardiomyocytes getting out and being a BSL-2 fluid inside the space station. So I find it hilarious that we can work 14 hours a day with no OSHA and no safety regulations, but like, oh my gosh, we can't have cell culture go anywhere. So we built this BSL-4 glove box to do the experiment in. And so here you can see this was a subsequent study where we actually, instead of the 6-well plates, we built these tissue chambers which had flexible posts on them. And this was actually an engineered heart tissue where we were looking at cardiomyocytes and fibroblasts and endothelial cells all together as an engineered heart tissue. And we were able to study contractility here. And so this is the paper from the first experiment that we did on board station. And hopefully the second experiment that we did a few years later is going to come out soon as well. Another thing that we're doing that's really cool is tissue chips, and that's actually with NCATS, and I'm going to go talk to them tomorrow. So they've had an incredibly successful program that's a collaboration between NCATS and NASA to fund 10 different investigators who are each looking at different tissue chips or different organoid systems on board the ISS. That program was so successful that we've actually started a second round of that, and using microgravity to try to study some really interesting physiology in these different organoid systems is really cool. So that's a short tour, and we could talk all day about all of these things that we're going to do on ISS, but I want to talk about where we're going in the future. And we are going to the moon, and we are going to Mars. And so I love this photo because for once, this is not a PowerPoint diagram of a theoretical spacecraft going to the moon. This is an actual NASA spacecraft, and you can see the moon there, and the Earth is beyond it. It's a farther distance. And so this was the Artemis 1 mission that launched. No people on this. This was an uncrewed test flight of the mission. It was incredibly successful. This went around the moon. We have our crew named for Artemis 2. That's going to do an Apollo 8 style mission around the moon. And then Artemis 3 will be a landing mission on the lunar surface. And so, of course, we're going to the moon, and I immediately think microbes. Because we prep our spacesuits, this is inside the airlock getting ready for a spacesuit, we prep our spacesuits in what is clearly not a sterile, clean environment. You're trying to reach the pump inside the suit to prime it, and you're dripping your eyelashes and your skin bacteria all over the spacesuit, and then we take it outside. So we go outside, we're doing a spacewalk, we're outside the space station. So my question is, when you go land on the moon and you set foot on the lunar surface, which is a sterile environment, it is the world's best controlled environment for doing microbial studies because there are no contaminating bacteria. My question is, what is the contamination that we bring with us? What's the contamination on the suit? And we can map this, right? Because we have a completely microbial-free environment. There's some tardigrades, one place that crashed into the moon. But except for that, the moon is sterile. So we're going to be bringing our human cells and our microbes, and I think this is fascinating to kind of study as a spatial temporal distribution. What is there? We don't expect too many live microbes to persist, maybe some spores, but I'm really interested in the contaminating nucleic acid that we bring with us, as well as the proteins. And so kind of doing a mass spec and a nucleic acid analysis in space and time as we go out from our lander would be super interesting. And this is going to be really cool just because we can't do this experiment on Earth, and to study what happens to the persistence of nucleic acid. And then also when we go to Mars. When we go to Mars, we are looking for life, and the worst thing in the world is to say, hey, we found life on Mars, and it's a little Staph aureus that somebody dropped on the spacesuit on their way inside. So we really need to understand, I want to build a census and build a spatial temporal map of what we are likely to bring with us to Mars. This helps us also understand how bad the contamination is. Is this something that, after a few days we can't detect the nucleic acid anymore, so we don't need to worry about it, or this is a really long persistence? And I had a white paper that's still not gotten funded where I think we should go to the Apollo site and go pick up their bag of poop that they left under the lander and see if we can find anything, but so far NASA has not bought into this study, so maybe someday. But when we're thinking about Mars, when we live on this planetary surface for the first time, the more I think about it, the more it actually resembles this really other remote, challenging environment that I've been to. Every equipment decision that you make has to be driven by power, data, mass. You cannot rely on any power structure. You can't rely on there being clean running water. So you design your entire experiment or your entire clinical system around the fact that there's just not a hospital and there's not a clinical lab and there's not a bench where you can put all of your equipment on. And that drives your design decisions in a very different way. And so one of the things, another project that I'm working on is trying to find small point-of-care clinical devices that we actually put together in a predefined logistics package where we think about things like cold chain, we think about things like consumables, we think about power, we think about the space that it's taking up and how we're going to transport it. Could that fit into a shipping container? Could that fit into a predefined launch container? And can we use that for something like space as well as disaster relief or medicine in a remote environment? All of these things seem so different, and they are so incredibly similar in terms of logistics challenges. So I want to bring this back to our beautiful planet. And this is one of the most incredible views that we have from space. I think it's my duty and my obligation. I feel incredibly fortunate, and I don't know why I deserve to be one of the humans that was able to leave this earth and look back and see it, but I want to share that with people. This is the least that I can do. And I think our thinking at NASA, a lot of people at NASA, they think about the rockets and they think about where we're going, and I think about where we are and where all of the humans are, and how can we use all of this engineering and the money and the technology that we're spending in NASA to benefit life back on Earth. And I really think understanding the human physiology, which as you can tell, each system I talked about has a disease parallel, understanding these restrictions and these forcing functions that make us design equipment and design procedures and design experiments in a completely different way because we assume no resources to begin with. I think that's some of the power of the work that we can do at NASA and the work we can do in the future. And I talking to all of you that you are doing incredible work, that I can see the parallels there, and I think there could be some really cool stuff to work on in the future. And so with that, I really want to thank the huge teams that went into some of these major experiments. So this is the sequencing experiment. Really, Sarah Castro-Wallace is the one that spearheaded this. And then Joseph Wu's lab at Stanford for the heart cells and the cardinal heart experiment. And so with that, I think we hopefully left some time for questions.
We built in lots of times for questions, so please start queuing up at the microphones. I cannot imagine people are not going to have lots of questions. I have plenty if you don't, but I just know there's going to be lots of questions. We'll start here.
Thank you. Thank you. Great talk. So as you may know, in NHGRI, when we celebrate the symposium, we always recapitulate the movies. And then last year was Barbie. And then, but today I just want to ask you, did you watch Sandra Bullock movie Gravity before doing the spacewalk? And regarding that, like, how did that affect your experience on that? And the second thing is, I think you responded that because you did almost 12 hours outside. And then it was on August 19 and September 1st, 2016. And how was your experience? In Mexico, we have another question is, in Mexico, we have a group of people that run on the mountains, and every time that they say hi to one another, instead of saying, hey, hello, good morning, they say, what did you dream? And then, what did you dream there while you were sleeping? Were your dreams awkward or different?
Thank you. Those are really wonderful questions. Yeah, so I want to get to the first one. So Sandra Bullock and Gravity. What I'm going to say about Gravity is it is a beautiful, artistic, like cinematograph. It's beautiful. I mean, the director's incredible. It's this one long shot. It is a piece of artistry. It does not really reflect the laws of science and physics particularly well. I thought it was real cute that somebody's like, I've trained for 6 months and I want a spacewalk now. Like, no. It takes 4, 5, 6 years. We also don't wear booty shorts inside the spacesuit coming out that little tank top, and we don't look good, and we don't have all of our eyelashes on. We wear a liquid cooling ventilation garment that looks like some long underwear. We're in there for 14 hours. We're wearing a diaper, and I always have snot on my face when I come out after that because you can't really blow your nose. So that's the Hollywood and the reality that split apart real there. In terms of movies that I actually really like The Martian up until like the last 15 minutes where it gets physically impossible. So that's a really fun movie to me. And then my favorite movie, and you guys will all appreciate this, is Gattaca, because how prescient was that in the 90s? And it's got both genomics and space as part of it. So in terms of some of your other questions about doing the spacewalk, yeah, spacewalking is incredibly difficult. It's one of the most dangerous things that we do besides launch or landing. And we train underwater in a pool. The spacesuits weigh about 300 pounds. The training is brutal. When we come out of that spacesuit, it's pressurized to 4.3 psi. So it's like being inside a basketball wearing oven mitts. There are bruises all over. The spacesuits that we have at NASA are not designed to fit women or small people. And so as a smaller person, I have to like throw myself against one side of the suit to get my arm in the hand versus the other. So I've never been in a bar fight, but I come out looking and feeling like I imagine what a bar fight would be after I get out of these spacesuit runs. So there is a lot of work that goes into this. When we're out there doing it, there's not a lot of room to think about what you're doing on the spacewalk because it is so dangerous. Every single movement that we make, we have to be careful. We do have a safety tether that attaches back to the airlock, but when we're going hand over hand, that's our only tether. So if you're going hand over hand and you slip and you don't make the next handrail and you come off the space station, you're coming off with a 185-foot tether at whatever velocity vector you slipped off at, and you were going to get to the end of your tether, come wrap around, you could put your head through a solar array, you could bash your head into the space station. You're completely out of control. And so we are thinking about every handhold and every movement, every single second that we are out there. That being said, it's type 2 fun. You come back in, you're like, that was great. You give each other high fives. It's the best time you ever had in your life. And then I was spacewalk branch chief, and I would have people call me after their flight, and I said, like after their first spacewalk, and I said, how is it? And to a T, scientists, engineers, fighter pilots who had flown in combat told me this is the most terrifying experience of my life. And I said, like, what was it about this? And they said, it's so dangerous, and we can't really train for it on the ground. And so the first time you're experiencing this in vacuum and with this fully pressurized suit and in the real space station, that you just haven't been able to practice for that in your life. And so we've started talking about that a little bit more in the astronaut office. It's the attention to detail and what you need to do is that level is the highest that it's ever been in my life. And so it's not fun when you're doing it, but it's real fun afterwards.
Wow, what an interesting, I feel weird asking another question right after that answer. But hi, I'm Kelly, I'm one of the postbacks in Genome. I actually wanted to go back and ask about this kind of like recycling of compounds and elements. Since you guys aren't recycling, like carbon, nitrogen, things like that, is that all like coming in from the food that is brought on board, and then also the hydrogen gas that is being used in the Sabatier reaction. Is that like are you guys carrying pressurized canisters of flammable gas up with you? Is that just seems very counterintuitive.
Yeah, great questions. So essentially the carbon is mostly coming from the CO2 that we exhale. So this great little, living redox reaction that we've got here is generating a lot of the carbon, but carbon molecules come in through the food that then we put into the humans. That we then breathe out. And then what we do is when we split the water in the hydrolysis reaction, we actually have the hydrogen coming from there. So we're not sending up canisters of hydrogen, but yes, super flammable, kind of a scary gas. There's a lot of systems, valves, secondary failure systems, and checks. NASA is also an agency that puts people on top of controlled explosions. So we are pretty good at containing things and doing the engineering to put all that together, but that's one of the things we pay attention to very, very closely is what's happening to the hydrogen on board. Great question.
Can I ask a quick follow-up? So I was sort of interested in this kind of like the water being the same water that has been there for decades, and I'm sort of unfortunately a former chemistry major, and so I was wondering like, is that actually the case? Like, given that all of those elements are sort of being reincorporated into different things in the body and then being excreted in various ways, like, have you like? Has anyone done an experiment where they have tried to figure out like how much of the hydrogen and oxygen that started out as water actually ends up as water?
That is a great question, and yeah, I think you can think about it as like the bulk system has the same mass of material. So that's maybe a better way to put it, because we do put new humans up there, right? And these humans breathe the oxygen and they make new CO2. And so it is not that the molecular, every single molecule has never left the space station, but that total mass of water only decreases by 3%. And that's on the US side. And so how we make up that difference, rather than shipping up water, because that's super expensive, we've stopped doing that. The only water we ship up is very special water that needs to be chemically controlled for the cooling system in the spacesuits. The rest of it we actually make up by bartering with our Russian colleagues, because they don't have a reclamation system for their urine. So as an astronaut, one of your jobs is to go down to the Russian segment in the morning and ask your Russian cosmonauts very nicely for a hot bucket of pee so that you can bring it back to the US side and recycle it.
You're not kidding.
I'm not kidding. It's so much fun.
Thank you so much. It's difficult to come off of the answer about the hot bucket of pee. Speaking of, I guess, glamorous jobs, you mentioned a bit about how much effort goes into just maintaining like the astronauts' physical bodies, as well as how long and intensive the experiments are, spacewalks, things like that. I was wondering if, it's kind of a two-part question, but if you could answer, how much of your day-to-day work can actually be attributed to scientific work versus just daily maintenance and things like that, and also thinking about in the future, thinking about long-term human habitation in space and on the moon, how to create a schedule that isn't so onerous on the astronauts, but able to actually create productive science in space.
Yeah, wonderful questions, both of those. So our time on the space station, I would say, is at least 50% science, if not more. So we have, these kind of 12-hour days. We do DPC to DPC, daily planning conference in the morning to daily planning conference in the evening. They're about 12 hours separated. And we do 2.5 hours of that as exercise, and then the remaining time we split up with these various tasks. And so a lot of time the tasks will be different scientific experiments, and we have some ongoing maintenance that we need to do on the space station. Now, if something breaks, like if the water system breaks, we slow down some of the other activities, and we need to go fix it. Because if the toilet breaks, nobody's happy. And like my first flight, one of the first activities I got was fixing the toilet, and I did too good of a job, and they're like, great, Kate's the toilet girl for the rest of the mission. So I got to fix the water recycling system the whole time I was up there, but it was pretty cool. I mean, this is an amazing system. So we focus on maintenance when we need to. If something goes wrong, other than that, we're doing preventative maintenance, but the day is more than 50% science, and then the rest of it is taking care of ourselves and taking care of the spaceship that we live in. And then, did you have another question?
Yeah, just thinking about in the future how to sort
Oh,
of
yes.
speed that up so that it's not so
Absolutely,
much.
and so this is one of the things that we're really working on. So when we go to Mars, number one, those systems need to be smaller, right? Like our water system's amazing, but it's 3 refrigerator size of water system. So trying to figure out how we can get that down into smaller, more portable systems, and then we also need to increase reliability. So right now, if that system breaks, I can get some spare parts sent up from Earth, not immediately, like every 3 or 4 months is when we can get a delivery, so we have some spares stocked on board. But we kind of have this idea that we can order more spares. Once we go to Mars, we're going to do our trans-Mars injection. We are coming home for 2.5 years, and whatever supplies we've sent ahead or whatever supplies we bring with us is what we've got. And so the systems are going to need to be designed for greater reliability. And so these are 2 things that can actually help if we're thinking about this for systems on Earth. Obviously, 3 refrigerator sizes is not practical for a water reclamation system. As we get this more portable, and then if you're going to set up a water reclamation system somewhere, you don't want to constantly having to have people fix it and supply it with parts. So we need to increase the reliability. So it's an engineering research driver, which is really cool.
Thank you.
Dave?
So we talk a lot, or we show these slides a lot of costs per genome. I noticed you didn't tell us how much it costs to do your, no.
I don't think
That's not my
I
question.
don't think we've added it up, but that would be interesting.
Probably best. What I was really wondering is, I mean, you all are just, engineering and ingenuitying your way through all these problems. Are there any problems just in doing the science that are just so stubborn that you just avoid those areas, or?
Yeah, that's a great question. So we have a hard time with any kind of scientific investigation that we want to do that has a big piece of equipment. Astronaut brains are so interesting. When we get back, when you do fMRI, we lose a lot of gray matter, but we also have, on the fMRI, you can see lower body sensing and movement lights up because we start to use our feet as sensory organs in a different way. I talked about the need to stabilize. You always have to hook your feet under a handrail, and you don't, like right now, I get to stand, and I'm not doing that much stability unless I'm moving. But on the space station, this and you're sideways and you're falling over unless you control your motion at all times. And so we hook our feet in and we control our motion with our shins. So your calves shrink and your shins get big, and it looks wrong. But so we're kind of doing this stuff all the time, and so it makes sense that your brain is actually really going to develop a lot of thinking in terms of using your lower body. Because you don't always look where you're putting your feet, so you're doing something else and your feet are tapping around and they can find a handrail to hook under, and then you automatically learn how to stabilize yourself in a completely different way. So I'd love to have an MRI on board. We just don't have that right now. So it tends to be big imaging things, large pieces of equipment that are too expensive. It doesn't mean we avoid it, it just means we still really want to do it and we get frustrated that we can't. So as things get smaller and more portable, that's when we can then think about bringing them to space in this really challenging logistic environment.
So, Kate, I'm going to inject a question because it related to Dave's joke about cost. So what I don't think totally came across in your talk is that when you're on the ground, you are constantly designing experiments and negotiating experiments that you then supervise that are done by non-biologists, typically up in the space station. That's a big part of your job when you're not up there. But you also are constrained and constantly making decisions about what experiments are going to be done because of the high cost per hour of astronaut time. Can you say a few things about the design of the experiments, the oversight of the experiments, and the cost of the experiments?
Yeah, absolutely. So I think when we design experiments on the ground, we think of space as free, and sad to say, people are cheap. And I was a grad student who ate ramen for 5 years, so I understand. We just don't get paid a whole lot. We don't tend to factor in the time of the researcher or the scientist. But on board, because the astronauts are operating the entire space station, fixing the equipment, and then trying to do experiments not just for, so I don't do my own research program. I assist with all these other people's research programs. And this is an international research program, so this isn't just the US experiments. These are all of our different international partners who are sending experiments. And so the astronaut time is incredibly expensive on board, and it's billed in 5-minute increments like our schedule is. And so anything that we can do to automate these experiments and do them in high throughput without human intervention is incredibly valuable. And so things that like Kate's Christmas wish list is automated sample prep, where I can take some clinical sample and I put it in and I maybe pull it out the other end and put it in a sequencer. Like I just want all that in a small machine that also doesn't draw a lot of power. So if anybody knows of one, please let me know, because I'm looking for it.
And the cost per hour of astronaut time?
It's $144,000 an hour, as NASA has calculated it with all of the kind of the budgetary things
So just
that we're doing on board.
postbacks who are out there, think about your year-long salary. If we get astronaut 1 hour, $144,000.
Yeah. By the way, we don't see a penny of that.
Yeah,
We're
of course.
just,
Want to tell them about your per diem you get when you travel?
Yeah,
The government bureaucrats here, they love to hear this.
So when you travel to space, they actually make you fill out travel orders, because as a government employee, I can't travel anywhere unless I'm under orders, so it has to go in the travel system. When you put in the travel system, it says low Earth orbit, comma, Florida, because it has to have a state, or the system won't accept state that you're departing from. And then you get back, and they said, well, you need to file your travel reimbursement. And I was like, what are you talking about? And they said, well, meals are paid for and lodging is paid for, but you need to file for the miscellaneous expenses, which is $5 a day. So that's what I make for going to space, is $5 a day. They're charging you $144,000 an hour.
Go figure. Next question.
I want to thank you for such an engaging presentation. As you touched on in an earlier answer, the spacesuits weren't made with women or people of smaller stature in mind. Would you be able to touch on what your experience as a woman in a male-dominated and primarily designed for men space?
Yeah, absolutely. So I will say, in general, working in the space industry, it's just not something that I think about very often. I do get this question a lot, like, what does it feel to be a woman in space? And I'm like, I don't know. I'm just me. What does it feel like to be a man in space, right? This is a ridiculous question. Like, you don't think about this. Where it does come into play is, you're absolutely right, is in this idea that equipment is designed for somebody of a different size. And so our spacesuits are more than 40 years old, and they were not designed for women. They were designed on a male body, and the original concept for spacesuits was going to be 5 different sizes, extra small through extra large. And NASA started out and they said, okay, well, we'll build a medium and a large spacesuit, and we'll see who we can fit. This is a man's medium, and I'm tall and broad-shouldered, and I don't fit in it. It's way too big for me as a medium. So they put everybody in it, and then they realized that some of the people wouldn't fit, and some of the bigger guys wouldn't fit. So they designed an extra large hut for the hard upper torso for the people that couldn't fit in the large, and they were working on designing a small upper torso for the people that the medium was too big for them. We went along in the design and the development process, and they're having a little bit of hardware issues when you design a smaller torso, like how do you kind of fit some of the components on there? And at one point, NASA said, this is getting too expensive. We have enough people that can do spacewalks, so we're canceling the program that's going to design the smaller suits, but we need to keep putting our money into the larger suits because we can't fit, most of the program was men, and we can't fit these people. I don't think that was the right decision. I think in hindsight, that was actually a terrible decision that we made. That we're still, it's having consequences 30, 40 years later as an agency. So when I came in, I was told, I wasn't treated differently. They just said, you're not going to be able to do spacewalks. Because women really don't do spacewalks. We have you guys control the robotic arm. You don't have the upper body strength for it. This was the line I was fed. Just like the pipetting in space, that made me real mad. It was a long and very difficult road to figure out how to make that hardware work for me. But I did do spacewalks, and when I came back after my first flight, the chief of the office actually sat me down and said, look, I want you to be the first female spacewalk branch chief, because we have a new class coming in that's 4 men and 4 women, and we want to change what we're doing about this. So we really changed a lot of the discussion from, you can't do this because you don't have upper body strength, to this is going to be harder for you because we did not design equipment. We have institutionally failed you, and we did not design equipment. And the really good news, part of the story and why I'm smiling, is because NASA has recognized that and righted that wrong, and we are designing new spacesuits now, and in the requirements that we wrote before we let the contract, it has to fit 1 to 99th percentile anthropometric range. And now that is not everybody on the planet. They took a military population study that was men and women, and said that we need to fit the 1 to 99%, and that's in all of these different anthropometric dimensions. And so I think that is a great recognition that we cannot succeed as an agency if we do not design hardware that works for everybody. So I think it's really cool going forward. I love working on spacesuit because it's a new thing that we're doing. We're entering a new era.
Anne?
Yeah, thank you very much. Wonderful talk. And as a budget person with no science, it was fascinating. But a non-science question. Who was your second celebrity phone call besides Francis?
Oh, you guys are going to make so much fun of me.
I know what you're thinking. It wasn't me.
I jumped it.
It was George Church. Yeah, I'm that big of a nerd. I know.
I told you she's a nerd.
I know. My crewmate called Ryan Reynolds and Blake Lively.
Right here.
I just wanted to say thank you for your presentation. I loved how you mentioned, what originally brought you into NASA, doing small, local, community-based things that kind of exposed you to this. And similar to the last question about being a woman in space, I'm actually really interested in some of the studies that have come out in terms of like female anatomy in space, and any kind of, like you talked a lot about the sort of things that happen as you go up. And so I wonder if any of that is different based off of male versus female, since they might have more data on the men.
Yeah, that is a great question. Actually, there's a paper published, I think, in 2021 that examines this and that takes a look at across all of these different physiological things. And there's a few things that are a little bit different in terms of this was focused on physiological risks and like why you might need to bring a mission home. And they found for the most part that there were not great differences. So for example, like the neurovestibular function that we see or the cardiovascular dysfunction, those don't segregate based on male or female. There's a few things that we are taking a look at in terms of some really subtle physiological differences, but you have to look real hard to pull those out. And so for the most part, these massive changes that are happening, the human body's sort of reacting the same way. And while there are some changes, and we're thinking about bone loss a little bit differently in women, there's not a whole lot. And so if you take a look at that paper, that's going to go into, and I'll see if I can find a reference and make sure that you get that. That'll go into what these little subtle differences are. But we had to kind of look to find them. So that was sort of a cool result.
Okay. Kate, thank you so much. I cannot extend enough appreciation for you coming and spending the day with us and giving this outstanding talk. And you are one of the coolest people I know, and your flight jacket is really cool. But I have something even cooler for you that actually I want you to open now and maybe switch into. It'll be much more interesting than this.
Yes! This is awesome. I'm so excited. I will absolutely, yes, I will absolutely put this on. Cool. Get out of NASA, get into NIH.
That's right. Here we go. We want to make sure we don't
untangle here.
get a wire.
There we go.
Okay, here we go.
All right.
We'll get you in this.
Now I feel like I'm in the cool kid club.
All right. There we go. Wait, there.
Okay. Thank you.
Okay, and with that, we will draw to a close day one. We'll see everybody back tomorrow morning at 9 o'clock. Thank you all. See you tomorrow.
Thank you.